Hi, it's Mrs. Shannon, and we're going to go through the structure and support Prezi that is located in your cell modules. And so I'm going to open it in full screen, and we're going to go through it pretty quickly, but make sure that you look at your PDQs so you know what to look for. PDQs are not just for the actual Prezi, though. These are questions to answer. If you don't find the answer in the Prezi, if you don't feel like you understand it quite yet, use your book. Or if these Prezis don't help you, you don't learn that way and you're more of like, I need to see the words to find the answers, use your book, use your test prep book, and use your other resources. The big questions here that this one's going to answer is, what does the internal structure of a cell look like? How do cells separate themselves from their environment? So what kind of features do they have? What kind of modifications have been made? How do cells communicate with the environment? We'll talk a little bit about that. We're going to focus a lot on cellular communication in a few more weeks. And how do cells communicate with other cells? So this last one especially, we'll touch on a little bit, and then we'll come back to. So if you recall here, cells have systems to process matter, process energy, and process information. So when we were looking at sort of life characteristics, and we looked at that Bozeman video on some of the characteristics, and we looked at that TED Talk, it talked about certain features that must be found in living things. And these 3 features that they need to be able to process any kind of element, matter, molecule that is inside of it. It needs to be able to process energy, so you're thinking metabolism, and it needs to be able to process information. That's the instructions. And from there, the information will be able to be passed on, inherited, and that's the reproduction. And then we haven't really talked about the communication, but cells respond to other cells. And that is something that we'll be focusing on. So inside the cell, we have the cytoskeleton. The cytoskeleton is what makes up the cytoplasm. It's a network of structural proteins that extend throughout the cytoplasm. So it's not just goop, it's organized goop. So when you learned about cytoplasm earlier on in the regular biology class, you learned that cytoplasm is just sort of like that gelatinous substances. It's actually organized into tubules, and that provides the network of the cytoskeletal system. The tubules themselves are organized depending on the thickness of it and what exactly they do. We have the microtubules, which will be the smallest. Well, actually, they're not the smallest. The microfilaments are. And then we have the microtubules and the intermediate filaments. And so they have different functions. As long as you can realize that these names, these types of structures help build that structure of the cytoskeleton, you should be in a good spot. I will tell you that whenever we talk about muscle contraction, we're going to talk about actin filaments and microfilaments. If you see like those, I think Gregorio videos or videos that I'll show you in class where it looks like organelles are sort of walking along long fibers, those are microtubules. They help with organelles being able to move within the cell, and that's the key here. And the microtubules are actually what provide that shape. And then we have our intermediate filaments. These also help with the shape, but they're more like load-bearing tubules. There's not as many of them, but they provide like the heavier support. So functions of the cytoskeleton: structural support, maintaining the cell shape, anchorage of organelles, and this anchorage will also tie into the regulation of the motility, being able to move around. So they don't just freely move around, the organelles, they move around because they're anchored to these microfilaments and microtubules, and that helps them move around. And also the movement of chromosomes during cell division. And so we'll look at how that works out later on in the semester. Okay, so here you can see a little bit more up close. Okay. And then here's like a 3D electron microscope view of it. So you can see the microfilament or microtubules and the red microfilaments. Another structural support piece is the cilia and flagella, and these are support pieces that relate to motility. They're extensions of the cytoskeleton, basically. And these extensions to the outside of the cytoskeleton actually allow for movement. And so here it's just extensions of the microtubules in an organized fashion. And the motion, what they do is they let cells go through cell space. Cilia is what's found all along the cell. They're like little short hair protrusions is what it looks like, and they help beat back and forth to move them. The flagella is the long one. Centrosome is another important component. It's only in animal-like cells, and it's sort of the microtubule organizing center. So when we go through cell division, this provides a very important component of being able to organize the chromosomes. The function of the centriole is the origin of all microtubules in the cell, and its major role in animal-like cell division. So that's inside the cell. That just makes up the foundation of the cell. At the boundary, we have the cell membrane, and we'll spend a lot of time going into the cell membrane. I have a couple more Prezis. The structure is often described as the fluid mosaic model. Fluid because it moves like a liquidish type substance, and mosaic because it has many different components. If you look at an art at a mosaic, it's different types of media that are put together to create some other artwork. And the same thing here. We're taking different molecules, putting them together, and they're going to work in a different way. Okay, if we were to cut open the cell, we would see the cell membrane going across. Inside of the cell membrane, if we were to cut directly in the middle, there's actually a bilayer. So you can see one layer can be split down and one layer can be split up. And it's composed of phospholipids, different proteins, also different carbohydrates. The functions of the cell membrane are the boundary of the cell, the transport of materials in and out of the cell. And this right here, we're going to talk a lot about in the next week or two. And then communication between the cell and the environment. And it's all going to center on the structure, the properties of phospholipids, so knowing the properties of lipids, and the interaction of these proteins. Okay, and so these are the common type of picture models that you'll see. We have our extracellular fluid on the outside, we have our nucleus on the inside, and we have our cytoplasm, which is composed of our cytoskeleton between. So here's everything that makes up the cellular membrane that we're going to be focusing on. Notice lots of proteins. We do have a lipid, cholesterol. It's going to help with the shape. We have lots of lipids, glycolipid. It doesn't show you in here, but each of this bilayer here is composed of phospholipids. So proteins, lipids, and then we have carbohydrates that kind of extend on the outside, and they help in cell signaling. But we'll get into that a little bit more closely. So this is a phospholipid bilayer. A phospholipid, you do need to recognize, it has hydrophobic tails. So you have these fatty acid chains that are coming down, okay? You have two of them. The thing is, at the top, you have a hydrophilic head. And if you remember, hydrophilic means water liking, water loving, and hydrophobic means water hating, water fearing. And so these parts are going to try to stay away from the water, which is why they're inside the membrane. These parts can interact with the water. They like water. And so this will be facing the outside into the extracellular space, or inside towards the intracellular space. Lipids with a phosphate attached to glycerol in place of a fatty acid chain, or in place of a fatty acid tail. And so what that means is you have the two fatty acid tails, and up here, instead of a third fatty acid tail, you have a glycerol molecule attached. Okay, the polar end is hydrophilic. It has a phosphate head. The nonpolar end is the hydrophobic fatty acid tails. It's called amphipathic, meaning it has both properties as both water-loving and water-hating. It spontaneously organizes in the presence of water to form a bilayer. And so when you guys were reading a question on liposomes and what that creation of those vesicles actually was trying to, what hypothesis it was actually trying to support. It has something to do with this because it spontaneously organizes. It does not need to be controlled. It is not controlled by scientists or anything else. It's able to self-assemble on its own in the presence of water. And the reason it does that is because when the water is around these phospholipids, they're going to organize themselves so the tails are away, and they naturally will arrange themselves in that pattern. Because of this difference here between hydrophilic and hydrophobic, it allows for a selectively permeable membrane. Only small nonpolars can easily move through, and that's because as soon as it gets to this layer, unless it's nonpolar, it's not going to be able to move because these are nonpolar. These are hydrophobic. And remember, like likes like. They stay together. So if it's hydrophilic, if it's a polar or hydrophilic molecule coming in, they are instantly going to be repulsed. They will not be attracted. These hydrophobic tails will not let them through. Another important component is cholesterol. I do want you guys to know the structure of cholesterol, the 4-ring shape, and what it does is it's a steroid lipid, and it acts as like a temperature buffer. It helps maintain membrane fluidity. So that way, if it gets really cold, it gives it some room so it doesn't lock up and become too rigid. And at the same time, it holds it together. When it becomes warmer and some of the bonds start to break, it will still hold that cell membrane together. Different proteins are in there. We have integral. Knowing integral and knowing peripheral, knowing what those words mean will be helpful. So if you do vocabulary flashcards, put these on it. Integral means that it goes through the entire layer. Peripheral ones are just found on one side. They're on the side. Peripheral means side. Like your peripheral vision for driving tests, you have to look off to the side. The results are membrane proteins are mobile in the cell membrane. And so what that means is that they can interact here and they can kind of move around. And so this is like on the outside here or on the inside. Actually, I think these are membrane proteins. These are peripheral ones. They can kind of interact and move around all along the top or outer extracellular space to it. There's other functions. So we have transport, which we'll be spending the next couple of weeks on. Enzymatic activity, we'll talk about down the road when we look at enzymes. When we look at chemical signaling and chemical cellular communication, we'll talk about the signal transduction pathways. We won't really do much with intercellular joining. And then we won't really do too much with attachment to the cytoskeleton. Integral proteins that span the bilayer regions extend extra and intracellularly. So here's our three-dimensional, you can see it's an alpha helix, but then the three-dimensional rearrangement. We have the N-terminus, remember NCC, and we have the other end, the C-terminus. That's generally how it falls. One's on one side and the other's on the other side of the membrane. You don't really need to know that in great detail. More so know that it's going to span the entire length. It does play a little bit of a role in signal transduction, being able to send and receive signals because of cellular recognition to the specific proteins. Okay, and I'm going to go ahead and stop here. I'll come back to glycoproteins and I'll pick up on the rest of it. I just don't want to go too long for one screenshot. Okay, so I'll talk to you guys later with the second part of this.